So we have a really wonderful session, and I'll introduce the speakers as we go. So the first speaker is Bernd Bodenmiller, and he's going to give us an overview of spatial proteomics to start with, and then he's going to go a little bit deeper of how we can use these methods to actually stratify patients. Bernd.
Okay, so thank you for the nice introduction, and thanks for inviting me here. So before I go into the different methods, I just quickly want to stress why is it so important to have spatial information. In our body, obviously, each cell is a function of multiple layers of external stimuli. So there's the direct cell-to-cell contacts that provide information, then there's signal processing across several cells, and then of course there are also systemic processes such as endocrine signaling. And also these signals are fairly complex. There can be proteins, can be lipids, it can be forces, and so on and so forth. So there's in fact not a single technology that can measure all of these spatial inputs onto a given cell. So if we now want to understand a cell in its context, we really want to have multiscale measurements where we can measure the molecules that are in between the cells that transmit information, the same ones within a cell to know about the pathways, obviously the cell phenotypes, the composition of a given tissue, and then where the cells are located, how they interact, and the overall 3D morphology that is relevant, for example, to understand whether there's a gradient of oxygen or nutrients. So here's a quick disclaimer before I'm actually getting started to talk about these different methods. First, I will only focus on single-cell resolved epitope-based methods. There's an enormous field of mass spec-based imaging, directly looking at proteins, lipids, and so on and so forth. But due to the sake of time, I cannot talk about this. And also, I will go through many methods, so I will have to simplify a bit. And also, in the moment, it's an exciting field. There's a lot going on, and so I really try to be fair and balanced. So there are different ways on how one can categorize this spatial protein imaging methods. And in my mind, one way to do it is to separate them between fluorescence and dye-based tissue imaging methods and those that use mass tags for tissue imaging. Now I will start with the fluorescence and dye-based methods, and I will start with those methods. And I will start with those methods that are close to immunohistochemistry that are using enzymes. Now, one important point I want to make here is you can see here some emission spectra of fluorophores, and you can see there's a lot of overlap among those fluorophores. And the result of this is that we cannot just measure 20 different markers at the same time, all coupled to a different fluorophore. But because there's this big overlap, the solution to this problem was to cycle the reporters, and I will go into how this is done. So the methods that are very close to the original immunohistochemistry approach, they rely on enzymes such as HRP on an antibody that's bound to tissue, and then the molecule is precipitated, providing a color that then can be imaged, and then either the antibody can be removed, and you come in with a different antibody, the same enzyme though, and then you need to precipitate a different dye of a different color. Or in this, that's a very long name, method called mix, here simply the dye is washed away, and then you can also do iterative cycles of imaging. And there's also one system, the OPAL system, that's commercially available. It can measure up to 7 markers sequentially, but in the end you overlay, so it's simultaneous in tissues. Now, what are the potential advantages and the potential challenges of these approaches? So the advantage of these pathology-like approaches is that you can use all the pathology equipment and the reagents. So it's very easy to do whole slide analyses. Also, you can do dozens of slides in parallel, because that's how these tissues are processed in the clinics. Also, you amplify the signal. It's all geared towards formalin-fixed paraffin-embedded tissues, which then opens to you the world of the big archives of the biobanks with millions and millions of samples. And already over 12 markers have been shown to be imageable with these methods. What are the potential challenges? So there's a fairly long turnaround time. Typically, the throughput is 1 marker per day. The dynamic range of immunohistochemistry is really low, so it's really not a quantitative method. Tissue can degrade. For example, here we try to do imaging mass cytometry after one of these methods, and you can see these are the nuclei, how they are destroyed after several rounds of this imaging approach. And we also found that there can be masking of epitopes. So then a second approach is based on fluorescent labeled antibodies. And here the antibodies are cycled. So the antibodies are bound to the tissue, they're imaged, and then either the antibody is removed or the fluorophore is quenched. Again, you come in with one or a few antibodies, and you go through cycles, through cycles, through cycle. And the first paper that really showed that nicely was from Schubert in Nature Biotech, where they imaged 90 antibodies on the very same tissue. And then there are many variants that have been published thereof, and a recent one, the 4i technique of Lucas Peltmans, what they actually found is that you have these fluorescently labeled antibodies, and you image them, they get cross-linked to the tissue, which require then harsh conditions to get rid of the antibody and signal, and they can add chemicals to overcome this, and this then results in much more gentle conditions to do this many cycles. And it also now addresses two critical points of this method. Because you have to cycle, the tissues can degrade over time. It's also a long-term process. And second, antibodies sometimes need a very long time to be stained on a tissue. And because you go through so many cycles, antibody binding becomes rate limiting. That's really the bottleneck. And for example, in this paper here, they have per round a 2 hours time frame. Now you can do 2 hours times 90 cycles. You're looking at a lot of time. Now to overcome this, recently there were several nice papers where they used DNA-labeled antibodies, where you can stain with all antibodies at the same time. And then what you're actually cycling are the readouts of the DNA tags. And one method is Codex from the Nolan lab. The other one is the Immunocyber that has an additional amplification step. And then you simply you detect, because you have unique DNA barcodes on a given antibody, you come in, you report for one antibody, you then remove the reporter, and you come in with a different one to a different DNA sequence, and so on and so forth. And the advantage here is that you overcome the bottleneck of antibody binding. It's much faster. And because it's also much faster, you have much smaller issues with antibody degradation. Now, what are the potential advantages and challenges using fluorescent-based multiplexing? So a big advantage is you can use any type of fluorescence-based instrumentation you already have around, and you can also use super-resolution microscopy. On paper, you could achieve an unlimited number of readouts. Practically, that's not possible, because what has been reported is 90, or over here, 56 markers that have been imaged on the same tissue. With the right equipment, you can also do whole slide imaging, and in the case of SABRE, you can also strongly amplify the signal. That also opens up the detection of low abundant markers. Now, what are the challenges? That's the cycling time, especially for the methods that rely on antibody cycling. It's tissue degradation. Some tissues are more sensitive than others, and then it's always an issue to do the cycling. And then we have to live with the world of fluorescence. So fluorescence has a low dynamic range, and then it's very easy to overexpose certain regions, so the information is not very quantitative. And especially in cases such as cancer, where the difference between lowest and highest abundance molecule can be fairly large, that is actually a challenge. And also there's autofluorescence that's shown up here. So there was no staining, it's just a kidney section that was imaged. And then there are also other issues, such as tiling effects, if you use just an out-of-the-box routine microscope, and you have to look at many fields of view that you stitch together. Then, now I have been covering the fluorescence and dye-based methods, but then there are also those methods that use mass tags for tissue imaging. And so here, all antibodies are simultaneously added on the tissue, and then each antibody of a defined specificity is coupled to a unique metal isotope of a defined mass. And then we can use a mass spectrometer to determine in a given tissue area the metal concentration and which metal isotopes are present. Then there are two variants. One is imaging mass cytometry, where lasers used to ablate the tissue to introduce it into mass spectrometer. And there's another method called multiplexed ion beam imaging. You all know it probably as MIBI, where you use an ion beam to generate secondary ions. And just recently, they published this nice paper in Cell, where they also use a time of flight readout, opening up the possibility to measure many metals at the same time. Now, what are the potential advantages here, and what are the potential challenges? So for both methods, there's a very high dynamic range. It can go up to 10 to the power of 5. There's no autofluorescence, not even in FFPE, since we do not look at fluorescence. We saw sometimes some background signal, for example, from patients treated with cisplatin, or that had contrast agents that also contain metals, and this we then see in some of the channels. Both methods have been shown to be highly reproducible. There's an extremely low signal spillover across the measurement channels. And in the case of imaging mass cytometry, we actually applied the whole tissue section, and this makes the method highly quantitative because we analyze everything. While in MIBI, you scan layer by layer by layer, and this offers you then the advantage that you can rescan the tissue, you can go in 3D with 20 to 30 nanometer increments, but then probably this affects a little bit the quantitativeness. Further advantages of MIBI is that you have a tunable resolution, I think with the latest system, down to 260 nanometers, and it can also make it much wider to the micrometer range, and it has a very high sensitivity as well. And for both, up to 40 markers have been shown to be measurable simultaneously. What are the challenges? First, you need a dedicated setup. You cannot just walk to a machine that you likely already have in your lab. There's not even theoretically an unlimited plex possible because we're limited to the isotopes that are used. In the moment, it's not possible to do whole slide imaging because the speed is too slow for those methods. And also, there are so-called tiling effects in MIBI. And for imaging mass cytometry, we have a nominal resolution of 1 micrometer that at least with the system we're using now, we cannot tune easily. And this resolution then for some questions, if you want to look at organelles, is simply not high enough. So now that I gave you this overview, I just want to say a few more things about imaging mass cytometry, and then give you an example on why spatial information is actually relevant in the translation or in the analysis of patient samples. In imaging mass cytometry, we have these metal isotope reporters on DNA probes and antibodies to measure transcripts and epitopes in the same tissue. As I mentioned before, with a laser, we introduce this into the mass cytometer, where we then do the imaging. Also here, a quick view on how those images look now. So that's a cell line that was stimulated with EGF, and you can see here for phospho-PLC gamma 2 how the signal is coming up. The resolution is also high enough to see, for example, cells that are mitotic. You can see here the different chromosomes and how they are separating. That's another example. That's a cell line, MDI-MB231, where we can also nicely see the Vimentin filaments or cells that are in late mitosis. That's over here. And also, when I did a direct comparison between immunofluorescence and imaging mass cytometry, I would claim that the data by now looks fairly similar, and we have a slightly lower resolution, as I just stated, but that really does not influence the analysis, at least we are doing that on a cell-based level. And the same is true if you look at tissues. So this was a proof of concept measurement of distant metastasis in breast cancer, and especially here, we look at bone, where then often with fluorescence you couldn't image because it's simply not transparent. We can still do this analysis with imaging mass cytometry. And by now the approach is fairly robust, which also means that we are part of different large initiatives where the technology is used as a core for the imaging. And also we are now in the clinical trial setting where we get samples and have to turn around the results in a 2-week time frame. And also this now is nicely possible using imaging mass cytometry. As for the data analysis, I'm really excited to be here because there are lots of smart computational people that I hope in the future will greatly contribute to analyze these data in a spatially resolved manner. And we in the moment do fairly simple analyses. We have a toolbox called HistoCAD where we can look at the images, we segment, we determine the cell phenotypes. Then we also implement an algorithm to determine the direct cell neighborhoods and cell-to-cell interactions. Then we can determine different areas, such as the tumor and the stroma. We can calculate distances and several other features. But I believe that's really very basic analyses that we are currently doing, given the nature of the data. And to further support the expansion of that, we now have developed a HistoCat version that originally was in MATLAB. We now have one that is cloud-based, where we also separated out the database, the processes, and up in the web, then we simply have a viewer. So we separate those two layers. And the great advantage is that now everything's highly flexible to implement new analyses. And most importantly, we can automate all of the workflows, which again is relevant once you start to look at hundreds or thousands of patient samples. Now, in the last few minutes, I want to give an example on why space is relevant when we want to stratify patients. So here in one study, we analyzed 352 breast cancer patients for which we had long-term survival data. Over here is the antibody panel that we analyzed with different cytokeratins, hormone receptors, immune context. We had cell growth markers, markers of epigenetic marks, cell death, and so on and so forth. And we imaged those across all of these patients. Then we determined the phenotype clusters. For this, we use a phenograph. And I will not go into details, but here on the x-axis are the markers that we use, the y-axis the different phenotypic clusters. We then summarized these phenotypic clusters into meta-clusters to have an easier biological interpretation and downstream analysis. And overall, in the end, we had 26 different cell phenotype clusters across all of these hundreds of patients. Then we started to look at those patients as a typical pathologist would do. So we gave a look at the luminal A patients, the luminal B, HER2 positive, and triple negative. So these are the 4 categories that are used in the clinics to decide on the treatment of patients. And if we restrict our markers only on those that are used in the clinics, we have a fantastic overlap compared to pathologists. And I don't show you here, but we have a really nice correlation. But now once you look at these phenotypic clusters across these different clinical patient classifications, you can see that it's really heterogeneous. And so this really shows that the current classification does not reflect the phenotypic heterogeneity. So we wondered, do we actually find relationships to clinical features such as survival once we actually look at these phenotypic clusters across of the patients? So here you see a plot of all these phenotypic clusters for every patient. We clustered according to that, and then we could identify 17 distinct groups, and then we looked whether those groups have striking features in relation to, in this case, survival. So here are the survival curves based on pathology classification of luminal A, luminal B, triple negative. These are those patients with the worst prognosis. And in this case, surprisingly, HER2 positive patients were the one with the best prognosis. So if we then look at our single-cell pathology groups, we can find about among the luminal ones that are related to those, one subgroup that really has an excellent prognosis. If we go to another group that's also hormone receptor positive, but with low expression, again, we could actually identify a subgroup that has an excellent prognosis. And even among the triple negative patients, there was one subgroup with an excellent prognosis, while all of the others had, as expected, a below average survival. And I didn't put the stars here, but those are also then significant compared to all of the other survival curves. And I will now not go into the details and try to explain you why we see these differences. Some are related to treatment response and not, but I really want to go into the aspect of space. So if we look at this group 1 over here, and the group number 3 here, if we look closer, then these 2 patient single-cell pathology groups are driven by the same tumor cell phenotype. But still, they have a different survival curve. Now, if you look closely at those images, there are just 2 examples. Here we have dense sheets of these tumor cells, and over here, they're much more sparsely distributed. So this made us wonder whether this distribution of many cells in groups, communities, whether that might show a relationship to clinical outcome. So we took this data, we looked at the phenotypes, we also looked at the neighbors, and then in this case, we also looked at cellular communities. And also, we started to look at gradients, and I have to say in these both cases, still an incredible amount of work to do on how to derive this information from these patient samples. And so what we did was very simple. We simply made a neighborhood graph where the node is a cell and the direct neighbors are connected with an edge. And we simply use Louvain to derive cellular communities based on these spatially resolved images. So once we look at these epithelial communities, we could identify across all of these 100 patients that there were 23 distinct communities. And so if we then look at those communities, you see, once you plot them at t-SNE, this very weird pattern or patterning. And if you look along this axis here, then it actually is related to the number of cells that are found in the community. So here, this is community number 16. You see small communities. Community 22, here you see the patches of the tumor cells are larger. And then in this community 14, the community patches are even larger. So here you have densely populated tumor cell sheets. If you then look at the immune cells, it looks very different because the communities become very heterogeneous. And also here then we could identify 29 of those across all of the patients. And also here each community has a different composition of these immune cell phenotypes. So now if we go back and just plot the tumor cell communities across the single cell pathology groups, and if we now go back to the example of group 1 and 3, then we see that in this group number 3, that has this much smaller patches, that's also 3, then we see that that's enriched in group 3, this community, but in group number 1, we have a different enrichment or absence of a different community that describes a much larger patch of these cells. So then we made a model where we looked at the grade, we looked then at this tumor communities and the microenvironmental communities, and we assessed whether we see a relationship to good and worse or bad prognosis. And then indeed we could find that frequently if the tumor had, although having the same tumor cell phenotype, once the communities are small, they have a bad prognosis. Once they have large patches of these communities, they have a good prognosis. There are other examples. Also the one that I've just shown you is actually over here and also over here. But what I want to stress here, that's only based on the tumor cell communities. And they're fairly homogeneous. Even though if patients are very heterogeneous in the tumor cell content, we found that communities are fairly pure with the tumor cell content. Rather, you will have in one patient different communities with different phenotypes, but each community contains mainly one phenotype. But then things are much more complicated than that. Because if we now look at the microenvironmental communities that are fairly heterogeneous, then we could also find here that in the case of triple negative patients, that over here, triple negative being the best predictor of bad prognosis, if you have certain communities, these patients can actually have a good prognosis. And that's also, so there are several of these complex communities of the stroma that we could identify that shift a patient from a subgroup that in the clinic says very bad prognosis into a subgroup that has a good prognosis. And that was just one small example and one little glimpse on all of what we did in this study. And so with this, I forgot that I added this slide. Because also now with IMC, we're doing 3D analysis, and you can see here, I hope it's repeating, you can start over here a micro protrusion of the tumor and then individual tumor cells. And so then we can look at spatio-temporal aspects within the tumor, such as the metastatic cascade. And with this I'm done. I greatly want to acknowledge my whole lab, and especially Jana Fischer and Hartland Jackson, who did this big project on the breast cancer analysis. And with this, I'm ready and I'm happy to receive your questions.
Thank you for a great talk. Questions? How do you validate your antibodies? Is it difficult?
So yes, it's a long and tedious process because to validate one antibody panel can easily take half a year or up to a year, depending on the markers you're interested in. But obviously, you need positive controls, you need negative controls. What is really helpful once you multiplex is you know which markers are typically co-expressed or must not be co-expressed in the same cell. There's a certain level of internal validation. To get your control samples, you can overexpress proteins, or you can silence, or you take tissues where it's known that they are there or not. So, but it's a long iterative process. And another important aspect is you want to achieve a nice signal to noise for your markers, especially or at least in the cohort that you analyze. And then we often have to go through many antibodies to find those that actually give us a solid signal to noise.
Cool story. Thanks for giving us this nice overview to start with. But of course, we're all interested in sort of leveraging the spatial aspect. So if I understood correctly, this nice story, first the association with survival that was based purely on the single-cell profiles, but then later if you use those communities, those sort of nearest neighbor graphs, you found better association. Was it purely based on the densities that were increasing? So if you just reduced all this information of the clustering with the nearest neighbor graphs to how dense cells are, would that give you already the same result, or is there more information, sort of maybe more subtle type of patterns involved?
So there's more information in there. On the tumor cell level, yes, it's more like a factor of fragmentation. In fact, that's also something pathologists look at. But if you look on the stromal cell communities, there's much more difficult. There's not the level of fragmentation. There is really which cells are actually present and how many, etc.
You don't look at this jointly also?
Pardon me?
You don't yet look at them jointly also, sort of interacting or something like that, close by in those networks?
So what we did for this analysis, that when we looked at the stromal communities, we just assumed that tumor cells are like one cell type to avoid some complications if you look at all at once.
Thanks.
We have time for one more question. Two more questions.
So thanks for your talk. And here. On the left,
yeah,
right.
okay, thank you.
So thanks for your talk. And so for the comparison of multiple slides, I think it would be critical to put all the slides in a comparable spatial to align the slides from different slides. So how did you do that to align the different slides to a comparable location?
So that's an excellent question. Basically, how do you compare multiplex measurements across hundreds of slides if you want to go through a large cohort or in a clinical setting? So for the study that I've shown you in the end, we in the end had a tissue microarray where all of the tissues were arrayed on the same glass slide, or actually it was few of them. And then you can do the antibody staining across all of the samples at once. And then also, that's how you avoid mostly the artifacts you can introduce by using many glass slides. But we have done a lot of validation on how robust and reproducible everything is. And one key aspect that introduces variability is actually the antibody staining step. So if you have to repipette the panel, that's when you introduce a lot of variability. And one way to overcome this is to lyophilize your master mix, or you can also store it at -80 degrees. And every time you get a new slide, you can use the same staining mix. We automate pre- and post-processing of the slides as well, which also helps to reduce this. On an instrument or technology side, the drifts that we get, or the variation that we introduce, they're really small. And I can show you like the slides where we looked at every channel across many patients, different operators, but that's not the driver for variability. Biological variability is much larger than what we introduce technically.
Dana.
So I have a scientific question and a meta question. The scientific question is, it seems like you're using one view per patient, and how much do you think that affects your survival analysis? And more meta for this community, you've shown that the technologies are already mature enough to do something with them, but still have many limitations.
So that's an excellent question.
And so so how do you think is the perfect balance between pushing the tech further to overcome some of the limitations you told in the first half and go forward in using them? Where do you find the percentage of balance? Those are my two questions.
So I believe that many of the technologies I showed you before, they're ready to address important biological questions. And each of these technologies has different strengths and weaknesses, and then it really is a function of the question that you want to address. If you're interested in subcellular distribution of proteins, then you should go for a technology that gives a really high resolution. If, like what I've shown you, we just look on a cellular level, and in any case integrate across a cell, then very high subcellular resolution is not really relevant. So it's really driven by the question. I still believe that while these approaches are now start to become useful to address biomedical questions, but even already in a clinical trial setting. I still think they need to be further developed to be much easier to use. There's a lot of expertise knowledge needed to do all of these technologies well. There should be much more automation, standardization. I guess an important topic here is also to develop standards to compare results across labs. So, and all of this is still absent. There's a lot of work to be done. Now, the first question that you've been asking about how many regions. So those regions were selected by pathologists. So there's a subjective input, but we actually got the same question from the reviewers. And so we analyzed a second cohort. We analyzed different regions of the same tumor to exactly address this. And the answer is that for most of these pathology groups that we define, actually one core is sufficient to correctly classify a patient. But then there is, I think, is a single-cell pathology group 6, 7, 8, so there's 3 or so, where it's really very diverse. And that's then the cases where you should image many more regions to then have a conclusive assignment of these pathology groups. So in the end, the answer is it depends on the patient, and it depends on the single-cell pathology group.
Okay, so thank you very much for a wonderful talk. Let's thank Bernd.
Thank you.
Thank you.